Hello everyone, today we're going to look at some of the important terms in biostatistics. First, we're going to look at the difference between the population and the sample. So the population is the collection of all individuals of interest. So it's the entire population, all of the people. Whereas sample is the subset of the population that is selected as the result of sampling. So we take the population, we take a sample from it, and then we get a sample. It's smaller. And why do we use samples? We use samples because they are feasible and their cost is low. So what does feasibility mean? It means that the research can be done on samples because the sample number is small, so the research can be carried out easily. And the cost is lower because only few people take part in this research. And here this figure is showing sampling, and here we have a population and a sample. And here the sample is representative of the population. Why is that? Because the ratio between different people is the same. For example, this category is 50 and above years old, and this is 40, this is 30. So we're going to take the same ratios. They're all like the same. Here 2, 2, and 2. Okay, so this is a representative sample. It should be representative of the population. Whereas here we have 3 different scenarios. Here is the population, and here are the samples. This is unbiased, so it's a good sample because the ratio between different categories is the same. 1, 1, 1, 1, okay? Whereas here, this is biased because we have 2 black, 2 red, 1 yellow, and 1 blue. So this is not objective, it's biased. And this one as well. Okay, we're going to look at 2 other terms, precision and accuracy. Precision is how close are the values from each other, whereas accuracy is how close is the value from the true measurement, from the actual measurement. If we have these 3 measurements, and this is the true one, we can say that these are precise because they are close to each other. It's 3.29, again 3.29, and this is 3.28. 3.29, 3.28. So they are close to each other, so they are precise. And they are accurate because they are very close to the true value, which is 3.3. If we rounded this, it will be 3.3. If we rounded this, 3.3, and this as well. Okay, we're going to look at this diagram. The first one on the left is not accurate because the values, the red spots, are far away from the true value, the central one. These are far away from it, so they are not accurate. And they are not precise because they are far away from each other. When I look at this one, they are precise because they are close from each other, but they are not accurate because they are not close to the central value. Let's look at this one. They are not precise because they are far away from each other. They are accurate because they are close to the center. Here, both of the conditions are met. They are accurate, close to the central value, and they are precise, close to each other. So that's all about accuracy and precision. Okay, now we're going to look at validity and reliability. Validity is the extent to which the instrument measures exactly what it should be measuring. So it's all related to accuracy. So the true measurement. Reliability, on the other hand, is all about precision. And it's the extent to which the results of a study are consistent with when the study is repeated under the same conditions. So if we repeated a study and we got the same results, our results are reliable. They are precise. Okay, now we're going to look at data and variable. Data is the values that we get when we measure a variable. Okay, and a variable is something whose value change or vary. Okay, so here in this example, we have the score and the 6. The score can differ. So it can be 90, 50, 60. So this is a variable, it's changing. And the 6 as well. It can be female or male. So it's changing as well, so it's a variable. Whereas the data is the numbers. This is the data, it's 42, 60, female, male, and so on. Okay, now we're going to look at other two terms, theory and hypothesis. What's the difference? A theory is a generalization about a phenomenon. It's something general. But the hypothesis is a proposed explanation made on the basis of limited evidence. This is the hypothesis. You have to know the difference. Okay, so here are two other very important terms, exposure and outcome. Exposure is a variable thought to predict an outcome variable. And outcome is a variable thought to change as a function of changes in an exposure variable. We're going to look at the example to understand more. Okay, so the exposure, let it be a medicine. Maybe a medicine for blood pressure. So this is the exposure. Our patients are exposed to medicine, so the medicine is the exposure. And then the outcome is the blood pressure. We are measuring the blood pressure. We are measuring it, so it's the outcome. It's changing, and it's the outcome. It changes as a result of the exposure. Okay, so you can link the exposure to what's called the independent variable. They are the same variable. They are the same thing, because the independent variable is something that you are giving in order to change something, and the outcome is the dependent variable. It's changing with the exposure. Keep this in mind. Okay, and that's all for today's lesson. I hope you enjoyed it. Thank you for watching.